First 2 minutes per introduction.
Welcome, Asghar. So we're returning. This is the third video today. My name is Brent Brown, and this is the social media group I started some time ago called Grow Your Own Maths and Science Like a Superhero. I'm welcoming Asghar from Park Bunair in Pakistan. Asghar,
Park British
botanist.
Park British School, Bunair.
My apologies. Thank you for correcting me. Asghar, it's a pleasure to be welcoming you.
Once again, thank you, sir. Today, inshallah, we will explain cellular respiration, and in cellular respiration, we will explain only glycolysis. First of all, we will define the respiration. So, the respiration is the oxidation reduction process that occur in living cell to break down food and release energy. Respiration is the oxidation reduction process that occur in living cell, during which food are broken down and energy is released. So, what are the importance of respiration? The purpose of respiration is to release energy stored in organic food molecule and ATP molecule are produced. ATP contains the right amount of energy available to the cell for the function. When ATP is broken down into ADP and phosphate. So glucose C6H12O6 plus 6 CO2, it will broken down into 6 CO2 plus 6H2O and energy will release. The respiration are of two types. One is called aerobic respiration and the other one is called the anaerobic respiration. Aerobic respiration is also known as cellular respiration. It is a type of respiration that need pre-energy. In aerobic respiration is completed. In aerobic respiration, organic food is completely broken down into carbon dioxide and carbon dioxide and H2O, and energy is released. The reactant of aerobic respiration are organic food and oxygen, while the product of respiration are carbon dioxide, water, and 36 ATPs. So this is the aerobic respiration which need pre-energy, while on the other hand, anaerobic respiration is that type of respiration in which incomplete breakdown of food occur and without the utilization of oxygen. Here, oxygen is not required for the breakdown of glucose, and it's complete in two steps. One is called the lactic acid fermentation, and the other one is called the alcoholic fermentation. Here we will only discuss glycolysis. The glycolysis is derived from the Greek word glyco means glucose and lysis means splitting. So glycolysis is the breakdown of 6 carbon glucose molecule into 2 3 carbon pyruvic pyruvate and net gain of 2 ATP. The site of glycolysis. As we know that the site of glycolysis is the cytosol of the cytoplasm, and it's common both in aerobic respiration and in anaerobic respiration. uh, uh, Step. In two steps, the glycolysis is completed. One step is called the preparatory phase, and the other one is called the oxidative phase. Are you getting the points?
Yes.
Yeah. Preparatory phase, it is complete in two stages. Preparatory phase. Number one, preparatory phase is the phosphorylation of glucose by two ATP molecule. The process start when glucose react with ATP molecule. Glucose is converted into 6 phosphate. The ATP transfer it phosphate ATP, the ATP transfer energy and phosphate group. ATP transfer energy and phosphate group to the glucose molecule, and forming glucose 6-phosphate molecule and itself converted into ADP. The glucose 6-phosphate is then isomerized into fructose 6-phosphate that react with another ATP and form glucose fructose 1,6 biphosphate. It forms fructose 1,6 biphosphate. the glucose 6-phosphate converted into fructose 6-phosphate, and the fructose 6-phosphate is then converted, reacted with another ATP and pump fructose 1,6 biphosphate. Fructose 1,6 biphosphate split into two 3 carbon molecule. One is called the PGAL and the other one is called dihydroxyacetone phosphate. They are both are interconvertible preparatory phase complete with the splitting of fructose 1,6 biphosphate into PGAL and dihydroxyacetone. Both are interconvertible. Here the preparatory phase is completed. Now, after the preparatory phase is the oxidative phase. Oxidative phase begin when two hydrogen atoms are removed from 3-PGAL and then transfer to molecule of NAD and NAD, oxidizing PGAL to PGA and reducing NAD to NADH. Uh, each molecule of phosphoglyceraldehyde transfer high energy transfer high energy to ADP, forming ATP molecule, and itself is converted into 3-phosphoglycerate and converted into 2-phosphoenol pyruvate. In the last step, two, three, 1,3-phosphoglyceric acid is converted into 3-phosphoglyceric acid. Here, two formation of 2 ATP also occur, uh, and the 3-phosphoglycerate is then converted into 2-phosphoglycerate. Here, 2 H2O molecule are formed. After that, 2-phosphoglyceric acid is converted into phosphoenol pyruvate, and in the last step, phosphoenol pyruvate is converted into pyruvic acid, and 2 ATP molecule are also here formed. So the net gain of ATP is here 2 ATP molecule formed. Two are used in the start. So the net gain in glycolysis is the formation of 2 ATP molecule. It's common both in aerobic and anaerobic respiration. Now, when the pyruvate are formed here, they will go goes before going they will into the Krebs cycle. They will pass to another reaction which is called the link reaction. Pyruvate does not enter the Krebs cycle directly. Before enter the Krebs cycle, pyruvate oxidize into 2 carbon molecule called acetyl group. This molecule is attached to enzyme called coenzyme N farmingo acetyl CoA. A coenzyme A consists of a nucleotide and a portion of the vitamin B. During this process, hydrogen is removed from the pyruvate, which is taken by NAD. NAD is reduced to NADH. Acetyl-CoA enter the Krebs cycle. This is called the link reaction because it links glycolysis to the Krebs cycle. So it is the glycolysis, and we will summarize once again it in detail. So glycolysis, glycomine, glucose, and lysis mean splitting. Glycolysis is the conversion of glucose one 6 carbon glucose molecule into two 3 carbon pyruvic acid and net gain of 2 ATP molecule. Glycolysis occur in the cytosol cytoplasm, and it completes in two phases. One is called the preparatory phase, and the other one is called the oxidative phase. Preparatory phase start with the I'm explaining the glycolysis, not the Krebs cycle. Glycolysis start with the preparatory phase, and the first step is the preparatory phase. It start with the uh, when the glucose molecule react with the ATP molecule, it form glucose 6-phosphate. The glucose 6-phosphate is isomerized into fructose 6-phosphate, and the fructose 6-phosphate once again react with ATP, and it form the fructose 1,6 biphosphate. Preparatory phase complete with the splitting up fructose 1,6 biphosphate into glyceraldehyde 3-phosphate and uh, dihydroxyacetone phosphate, and both of them are interconvertible. Now the glyceraldehyde triphosphate, from here oxidation phase will start. Glyceraldehyde triphosphate will convert into 1,3-diphosphogylyceric acid, and the one 3-diphosphoglyceric acid will convert into 3-phosphoglyceric acid. Here 2 ATP will form, and 3-phosphoglyceric acid will convert into 2-phosphoglyceric acid. Onward, the 2-phosphoglyceric acid will convert into phosphoenol pyruvic acid, and the phosphoenol pyruvic acid will convert into the pyruvic acid, and here formation of 2 ATP will occur. So it's a glycolysis. Enough?
Thank you, Asghar. I'm just going to go back to the beginning because it's quite a complicated chemical cycle. Um, I'm just going to go back to what happens where, because this is common in animals and plants. Let me just find it. And now it's important to remember that the different parts of all these cycles occur in eukaryotes and cytoplasm or in the mitochondria. So we were talking about glycolysis. Asghar very well explained that, occurring in the cytoplasms of both eukaryotes and prokaryotes. Asghar, thank you very much for joining me.
Yeah.
As ever, very well explained. Thank you for helping me along with that.
Yeah.